All right, so gene expression occurs in two steps, transcription and translation. So these two things will occur in different places for prokaryotes and eukaryotes because prokaryotes don't have specialized organelles to take care of for these different functions. Eukaryotes do. So in eukaryotic cells like yours, there's a nucleus, right? And the DNA is housed inside the nucleus. So I like to think of the nucleus as the Library of Congress. It contains all of the literature, but you can't take the literature out of it. It has to stay there. So all of the literature is there that you could possibly need. You cannot take the literature out. So that's sort of a problem in eukaryotes because how are you then going to translate that into proteins? Well, photocopies are made in the form of RNA. So that's the transcription part. When you transcribe something, you're copying it verbatim, right? Like if you're in a court hearing and there's some person typing in the corner, they're transcribing the court proceedings, meaning they are typing word for word everything that is said. Okay? So, and if you're transcribing something from your textbook, it means you're reading your textbook and copying it to your document. Right? So that's transcription, it's copying verbatim. RNA, we're going to talk about just in the next slide. RNA is almost the exact same molecule as DNA, except that it's shorter. There's a couple differences, we'll cover them. But it's essentially the same molecule. It's a nucleic acid, it's got pretty much the same letters. And so what we do is transcription is making a photocopy of some of the literature in the Library of Congress that you can take out. So literature can't leave the Library of Congress, but anyone can go in, make a photocopy of relevant material, and take it out and use the photocopy to do whatever work it is they need to do. So RNA is a photocopy, not of the entire library. No, it's not a copy of all of the DNA, not the entire library. It's a photocopy of a small segment that needs to be used, one gene. So the cell wants to make one protein. It's like, I need more of this enzyme. I need more of this aquaporin, this portal, this transport molecule. I need more defense molecules, whatever it is that the cell needs. The cell needs this, it needs to build more of this one protein. So some signals go into the DNA, into the nucleus, find the correct gene in the library, in the DNA library, open it up, make a photocopy of it in the form of RNA. The RNA photocopy of the gene can leave, and that's where it gets and it's that photocopy, that RNA photocopy, that gets translated into protein. Okay. Now, so the RNA transcript can leave the nucleus and go to the ribosomes. So these are ribosomes. Remember, ribosomes are the protein building factories. They read the genetic material in the form of RNA and build a protein from it. So DNA to RNA to protein. That's the central dogma of biology. DNA to RNA to protein. The RNA is the middleman. Okay. Now, in a prokaryotic cell, there's no nucleus. So this all happens in one fluid string. The RNA photocopy isn't even off the press before it starts getting translated into protein. Okay. That all just happens in the cytosol, right? All right, so differences between RNA and DNA since we're covering it. So remember, DNA is this double helix. RNA is always single-stranded. Okay. It can loop up upon itself and make fancy shapes, but it's still single-stranded. It's also short, so like the DNA in our cells are 6 feet inside each, 6 feet of genetic instructions inside every cell. That's a lot of material. RNA is short, right? It's a photocopy of one single gene. So they're the length of one gene. DNA is all the genes plus all the junk DNA. RNA is the length of a single gene. And then another difference, RNA stands for ribonucleic acid, DNA stands for deoxyribonucleic acid, right? Ribonucleic acid, that's down here, deoxyribonucleic acid. They have slightly different sugar molecules, okay, that build the two of them, slightly different sugar molecules. And deoxyribo means that there's one less oxygen on the sugar that makes they're both 5-carbon sugars, but the deoxyribo has one less oxygen on it. Incidentally, that one less oxygen makes the sugar in DNA less reactive, more stable than the sugar in RNA. And that's why when you dig up woolly mammoths, we don't read their RNA because the RNA breaks down because it's much more reactive with the environment. It tends to break down a lot more rapidly than DNA because DNA is missing that reactive oxygen group. So DNA is a much more stable molecule. It persists at crime scenes. It persists in frozen tissues of woolly mammoths, you know, so we can really, like, RNA is going to be far too damaged to get any useful information out of it. All right, so the differences we've covered is RNA is single-stranded as instead of double-stranded DNA. RNA is short, the length of a single gene. DNA is the entire library, so it's very long. RNA and DNA have different sugars, right? Ribose versus deoxyribose. The last difference is their letters are a little bit different. So both of them have cytosine. Their letters, by that I mean their nitrogenous bases. So both of them have cytosine, both of them have guanine, both of them have adenine, but they don't both have thymine. So RNA replaces thymine with uracil, and you can see that these are almost identical molecules, right? There's like a methyl group missing on the uracil. So they look almost exactly the same. So 3 of the nitrogenous bases that make up the genetic code are the same in RNA and DNA. One of them is different. The thymine is replaced by a closely related molecule called uracil. All right, let's go over transcription first. So this is making a photocopy, transcribing verbatim from one kind of nucleic acid to another kind of nucleic acid. So in transcription, you have this enzyme. Remember, an enzyme is a protein that catalyzes chemical reactions. So this is an enzyme, and its name is Rna Polymerase. So you might remember what a polymer is that we just covered in a previous lecture for this chapter. A polymer is a long molecule made out of repeating subunits. ACE, anytime you see ACE at the end of a word, you're talking about an enzyme. RNA polymerase is an enzyme that builds the RNA polymer. So its job is to unwind the DNA to read it. So it unwinds, you can see that it's unzipping. It moves down the DNA molecule, it unzips it, it reads just one side, and as it's reading it, it builds a complementary RNA strand. Okay, so when we were talking about DNA, we talked about complementary base pairing, remember? Complementary base pairing, because remember, A's bind to T's, G's to C's. Well, that's useful when the cell needs to build a new DNA strand. So when the cell's building a new DNA strand, it separates the 2 DNA strands, and an enzyme called DNA polymerase is going to travel the 2 strands, reading one and building the complement. The same thing happens here. The 2 strands are unzipped. One side is red, the side with the gene is red, and a complementary RNA strand is built because complementary base pairing still works here because the letters are almost exactly the same, right? So here, anytime you see a G on DNA, a C is going to be placed in the RNA. Anytime a C is in the DNA, a G will be in the RNA. So C's match with G's. If there is a T in the DNA, RNA polymerase is going to place an A because T's match with A's in the RNA, the new RNA strand that's being built. The only difference is when there's an A in the DNA, remember RNA doesn't have, go back to this slide, RNA doesn't have T's. You know, A's match with T's. Well, DNA has T's, but RNA doesn't. It has U's instead. So anywhere you would have put a T for a complementary DNA strand, you just change that to a U for RNA. Okay, so that's the only difference, but there's still complementary base pairing rules that work here. And I'll add, you know, the reason that these molecules, I'll just go back a little bit, the reason A's only match with T's and G's only match with C's is because if I go way back here, you'll see that, you know, some of these nitrogenous bases can make 3 hydrogen bonds and some can only make 2, right? So these ones make 2 hydrogen bonds, these make 3. So A's and T's can make 2 hydrogen bonds, C's and G's can make 3, so they match chemically with each other. Okay, so it just has to do with the bond structures that they can form and their size as well. They have to be complementary in size. All right, so where were we? Yes, okay. So RNA polymerase. It does a few things at the same time. It moves along the DNA strand and unzips it so that one of the strands can be read. It's going to read the side that has the relevant gene, and it's going to build a complementary RNA strand from the DNA strand using complementary base pairing rules. Also, the strand of RNA that's being built is going to be anti-parallel to the strand that's being read. So let's look at this in letters just so it's not so abstract. Okay, so let's say that this is the DNA strand, the DNA, a segment of DNA up here. Okay. Now, there is going to be a promoter sequence that the RNA polymerase can read. And the promoter sequence, that's where a promoter sequence is a strand of, I don't know, like 25 to 50 nucleic acids long. I forget how long it is. But it's a segment of DNA that's pretty long, and it tells the RNA polymerase, bind here. Here's the start. When it sees that, it's going to bind. And that's not in here. I'm not going to, it's very long, and I don't have it memorized. I don't expect you to memorize it. Just know that there's a promoter sequence that tells RNA polymerase this is the start of the gene. So it'll bind to RNA polymerase. And whichever strand had the promoter is going to be the template strand. The template strand is the strand that's being read. It's the template for RNA polymerase for building the new RNA strand. So instead of a promoter sequence, I'm going to do a start codon, which is 3' TAC. So we can look along here. No 3' TAC. So this is going to be our template strand. 3' TAC, I'm not going to worry about that, you knowing that so much. So don't worry about 3' TAC. So this bottom strand is going to be the template strand. Okay, imagine there's a promoter sequence over here, so RNA polymerase binds here, and it starts reading 3' to 5'. Remember, we said that there's directionality, so the RNA polymerase finds the promoter here, and it travels from the 3' direction to the 5' direction. It goes 3 to 5. And then it's going to build a complementary strand. So wherever it saw a T, it adds an A in the RNA strand that it's building. Wherever it sees an A, it's going to put a U, because remember, U is the stand-in for T in RNA. DNA has T, RNA has U. They're basically the same molecule, just, for some reason, that's just how it works, okay? So U's and T's, they take the same role. Okay, so T's to A's, A's to U's, which is RNA's version of T, C's to G's, G's to C's, A's to, again, U, T to A, C to G. So that's the complementary base pairing rules that we talked about before. And also you'll notice it's anti-parallel, so the sugar phosphate backbone of the RNA strand that's being built is going to be backwards relative to the sugar phosphate strand of the template strand that's being read in the DNA. So the enzyme is reading 3' to 5', it's building 5' to 3'. Okay? So this RNA strand, it's not a mirror image. It's the nitrogenous bases are complementary using the complementary base pairing rules, and the sugar phosphate backbone is going to be anti-parallel or backwards compared to the sugar phosphate backbone in the DNA template. So you'll notice here that only one of these DNA strands is being read and the other one is being ignored. Now, I always get the question, is this strand good for anything? Yes, elsewhere. There are some genes that run in this direction, and there are some genes that run in this direction. And they're going to be at different places in the DNA. Sometimes they overlap. So it could be that there is a gene that runs on the top strand, from 3' to 5', and then stops here, and then this one continues forward. Right, on this side. So there are genes encoded on both strands, but they're in different places. Maybe they overlap with each other, maybe they don't. Okay, so but both strands, but for this, it depends. The cell's only making one protein at a time. So it's ignoring this strand for now. It doesn't mean that the strand has no useful information anywhere on it, and it's all at 6 feet, right?